Hi, how are you? According to a study linked below, gene expression, autoimmunity, small fiber neuropathy, and neuroinflammation have been suggested to be involved in the pathogenesis of the disease fibromyalgia. Today, I'd like to talk about neuroinflammation. Neuroinflammation doesn't work the same way inflammation does in illnesses like arthritis, where the fingers are inflamed. Instead, it causes neurological problems that lead to neurological symptoms. And the research on neuroinflammation is what researchers believe is the potential cause of central sensitization in fibromyalgia, which is why we have a heightened pain response. And just so you know, it's the afternoon, so puppies are going to be bouncing around. Abnormal neuroinflammation was found in a 2021 study on fibromyalgia and complex regional pain syndrome. Specific brain areas found this inflammation in such areas as the primary motor cortex and primary somatosensory cortex and other brain areas. Identifying these regions may contribute to improved diagnosis and the development of effective medical treatment for fibromyalgia. Well, that's what we all want. In January of 2015, researchers used a PET scan and an MRI in a study to show that people with persistent low back pain had markers of glial activation, which was correlated with a cytokine called IL-6 and a translocator protein called TSPO. So this was the first step in recognizing chronic pain and glial activation. Several studies have confirmed this. It's known that chemokines, a class of cytokines called fractalkine and IL-8 in the cerebral spinal fluid are elevated in fibro patients. They're implicated in neuron-glial communication and relate to chronic central sensitization and pain. Other studies have shown this same glial overactivation that results in low-grade neuroinflammation. That's the brain and spinal cord, that is characterized by BDNF, which is a gene found in the brain. Also IL-1B, a pro-inflammatory cytokine, and TNFA, another inflammatory cytokine. BDNF has been shown consistently in fibromyalgia studies to be altered as early as 2007. In 2017, a review of previous studies completed, mostly on animals, found that glial activity might be triggered by severe stress exposure and/or sleeping disturbances in chronic pain development. And I've had both in my lifetime. In 2019, a PET scan, MRI, and radiologans were used to determine the glial activation that occurs in the brain of fibro patients. This was a human study. TSPO, that translocator protein, is known to be low in healthy individuals, but is widely upregulated in microglia and astrocytes under inflammatory conditions. In this study, TSPO was measured in fibro patients and found to be elevated in several brain areas compared to healthy individuals. And this correlated to fatigue. This supports neuroimmune glial activation in fibropathology. And this also shows that there is relevance, as opioid-induced hyperalgesia is associated with glial activation. This was the first study to show direct evidence of neuroinflammation in fibromyalgia and the method that inflammation occurs. I'll talk a little bit more about the opioid-induced hyperalgesia in another video. Glial activation happens when an altered immune cytokine becomes inflammatory. It's considered innate, meaning you're born with it. So we are born with an innate tendency toward glial activation under certain circumstances. Glial activation is a normal, adaptive, defensive mechanism that can contribute to handling acute stress, limiting tissue damage, and restoring homeostasis. Now, you noticed I said acute stress, but prolonged stress is another story. So when this process malfunctions, it can turn into the primary pathogenic element. In animal studies, glial activation was shown as a key contributing factor in persistent pain. Glial activation can produce cytokines such as TNF-A and IL-1B that are thought to play a role in chronic pain and induce central sensitization and neuropathic pain. In that low back pain study, glial cells were found to activate IL-6, IL-1B, and TNF-A. These cause immune reactions in response to infections and tissue injury. And they have discovered that TSPO, a protein, is a marker of activated glia cells in neuroinflammation and may become a target for therapeutic pain. Don't you just love biology? So now we understand that glial overactivation has been shown to become activated in fibromyalgia and believed to be the contributing factor in neuroinflammation. So one final thought. Our brain's gray matter changes over time with fibromyalgia. Some of you have probably heard this before, but I haven't talked about it here on this channel. Chronic pain patients with cortical gray matter alterations observed with anatomical magnetic resonance imaging, or MRIs, showed reduced regional gray matter volumes are often interpreted to reflect neurodegeneration. But this hasn't been tested in regards to fibromyalgia. One interpretation is that the decreased gray matter is due to reduced blood flow in the thalamus. The regions affected are the bilateral insula and the ACC, which are pain processing regions. The longer a person has fibromyalgia, the more decrease in gray matter was found, but more research is needed in this area. So with all of this research that's taking place, I wholeheartedly believe they're getting to a place where they'll resolve this. At least that's my hope and prayer. I hope this all makes sense to you. I send you gentle hugs and support. Love you. Heightened pain response. Specific and other and identify the specific brain areas found. Now I just overactivization. Glial overactive. Which is a bit radiology. That translator functions and tissue injury.